It's my pleasure to introduce to you our speaker for today, Dr. Robert Potter. Dr. Potter is an Assistant Director of Infectious Disease Diagnostics Lab at CHOP. He also serves as the Director of Outbreak Investigations for Center for Microbial Medicine, also at CHOP. He is also an Assistant Professor at Department of Pathology and Laboratory Medicine at University of Pennsylvania. And Dr. Potter graduated from University of California, Berkeley, then completed his Ph.D. in molecular microbiology and microbial pathogenesis from Washington University School of Medicine in St. Louis. He then became a clinical and public health microbiology fellow, also at Washington University School of Medicine. His work was recognized by several awards, one of them being the Paul E. Stranyard Young Investigators Award from Academy of Clinical Laboratory Physicians and Scientists. He was also recognized by the American Urologic Association Research Scholar Award. And Dr. Potter is devoted to genomic and functional analysis of medically important bacteria, viruses, and fungi. He also contributes to clinical oversight of the Infectious Disease Diagnostics Laboratory, and he will be speaking on his exciting work today, which explores how viral whole genome sequencing is transforming infection prevention and control efforts in hospital settings. And without further ado, Dr. Potter.
All right. Thank you so much for the introduction, Susan. I'm really excited to be here and to share our preliminary work on identifying intra-hospital Norovirus GII transmission. My full title is a bit aspirational. Right now, most of the talk is going to focus on identifying. If I was to give this webinar a year from now, I would hopefully have a lot more additional information on how we can use this sequence data to interrupt transmission and protect patient health. So it's an unfortunate reality that oftentimes individuals when they're in the hospital are at increased likelihood of infection. And we call those hospital-acquired infections. This infographic describes many of the different metrics that indicate how bad hospital-acquired infections can be, both from an individual patient standpoint, but also a larger system-wide level. And so it's really important that as hospitals, we're doing everything possibly can to ensure that our patients are safe when they are admitted for care. It is actually surprisingly easy for microbes, and in this slide, we're using microbes broadly to mean both viruses, bacteria, and fungi, to go from a colonized patient who may be having an infection, or maybe is just colonized and is not overtly infectious, into a susceptible patient who is admitted to the hospital for a completely unrelated reason and then gains an infection in the hospital. And the primary mode of transport is either human-based, so via contaminated individuals who are working in the hospital or visitors or parents of that patient, and then also on contaminated medical equipment. Some of this medical equipment might be items that are used in different patient rooms, and also on contamination of the rooms themselves. And that is why hand hygiene, environmental decontamination are so important for how hospitals are able to stop infections from occurring inside of their walls. And the very first thing we need to do to stop hospital-acquired infections is to really recognize when they occur, which is not as easy as it sounds. Is this scenario where we have a disease X and we have 4 patients, patients A, B, and C. If disease X is really rare for this community, and on day 0, we see patient A become PCR positive for disease X, and then on day 1, patient B, patient C then also become positive, and finally on day 2, patient D becomes positive. If all these patients are on the same floor, and this is a rare disease, we really don't need additional tools to say this is likely an outbreak, and we need to do a specific investigation and deep dive to learn what led the transfer of disease from patient A down to patient B, C, and D. But it's a bit more complicated when we have a disease that's common in the community, for instance, norovirus, and when hospitals are very large, and so patient A may be on the same floor as patient B, but maybe that's a different floor than patient C and patient D. And so although we might have the same timeline of events where initially patient A is positive, then B, C, and D, because disease Y is common in the community and because there is now spatial separation of patients, it's less clear if this is actually an outbreak or is this just a representation of disease Y being common in the community and the sickest kids will then get admitted into the hospital. We're fortunate now doing science and medicine in the sequencing era that we have this phenomenal technique that gives us a molecular fingerprint that'll help determine if infections are caused by the same exact strain of pathogen. And again, we're talking about viruses for this talk, but the same technique can be applied to bacteria and fungi. Using the sequence information, we're able to really unequivocally say whether or not two organisms are the exact same or whether they are different from one another. And with this information, we're able to then feed into our infection prevention and control algorithms to say whether or not this is actually an outbreak or whether or not this is just diseases coming in from the community. CHOP has had a long history with combating norovirus GII as an occupational health threat. I'd like to draw attention to the date of this article about 11 years ago that describes a particularly bad outbreak that affected the staff caring for our patients. The Northeast region of the United States, which is where CHOP, the Children's Hospital of Philadelphia, is based, has a relatively high background rate of norovirus that I sort of attribute to this being primarily one of the most industrialized regions within the United States. This is a local news infographic that described a year ago about how 15% of all tests for noroviruses were positive within the Northeast region. Norovirus is also really big right now. This was an article from the New York Times in December that was then updated in January, describing that norovirus levels are really the highest that they've been in the post-COVID era, according to the CDC. And just as some virology, norovirus, especially because I primarily am a bacterial whole genome sequencing individual, and so this is my first foray into viral sequencing, and so norovirus is a really nice virus to get started. It's a single segment. Some viruses like flu are multi-segmented. It's about 7.5 kilobase pairs. Something like SARS-CoV-2 is 30 kilobase pairs, so norovirus is a bit smaller. And norovirus encodes 3 genes. There's this blue gene, which is the polymerase and some additional immune regulating genes. And then these green genes are the capsid. And the capsid is what forms this extremely tight protein coat that allows norovirus to survive on hospital surfaces, sometimes for weeks to months at a time while still remaining infectious. And it's what also allows the norovirus to really survive attempts at hand hygiene. So that's why it's really important that we use soap and water rather than alcohol-based hand sanitizers, and also environmental decontamination. There's whole special protocols for cleaning rooms for norovirus in patients compared to other organisms. This protein coat just creates the perfect shell for the norovirus to survive in the community. And because it's so good at surviving, it's able to cause these large hospital outbreaks. Norovirus is a genetically diverse viral genus. It's composed of species that can infect humans and other animals. For today, we're focusing on this G2 or genogroup 2. It is able to infect both pigs and humans, and it's the primary cause of hospital outbreaks, although we sporadically see human disease from some of these other norovirus strains. So our goal is to use whole genome sequencing on norovirus G2 samples to aid infection prevention and control measures at CHOP. Our approach is to use full-length PCR amplification and Oxford nanopore technology sequencing. And our hypothesis is that we will identify spatially temporally linked outbreak clusters with less than 10 single nucleotide polymorphisms, or SNPs. And this effort would not be possible without a new enterprise at CHOP, the Center for Microbial Medicine, that opened in July 1st, 2024 as a center for emphasis. The goal of the Center for Microbial Medicine is to really use next-generation sequencing technology, both against individual pathogens and the microbiome, to develop tools that will allow us to provide better care for our pediatric patients. And so I'm fortunate that next-generation sequencing for infection prevention purposes is a big component of this leveraging sequencing to improve clinical decision support within the Center of Microbial Medicine. There are two wings of our study. We have a retrospective positive viral samples that I was very fortunate to inherit from previous directors of the IDDL. We have samples dating back through 2019 through 2024 that we're able to use for our validation and then analysis pipeline. And then as of December 2024, we are now performing prospective sequencing for the hospital 2 times a week for infection prevention and surveillance purposes. So we're starting with samples that we know are positive for norovirus G2 from the IDDL's laboratory developed test PCR assay that looks for the most common causes of viral gastroenteritis in children. If we were to extract those samples without the clinical laboratory performing that, that would cost about $11 per reaction and takes about 2 hours. We're using this on the QIAGEN EZ2 viral RNA extraction system. And we are then taking that viral RNA and we're doing a first strand cDNA synthesis and then a full length PCR using QuantaBio reagents. So we use the Q-Script UltraFlex kit, which takes about 30 minutes, and then the Replica Hi-Fi Tough Mix, which takes about an hour. And initially, when we were developing this assay, we were quantifying the PCR amount of nanograms per microliter using qubit high sensitivity assay, but we really stopped that because it doesn't determine exactly whether or not the assay will work for sequencing. So once the PCR product is complete, we are sending it to Plasmidsaurus for overnight Oxford Nanopore technology sequencing, which costs about $20 per reaction and is pretty consistently taken about 12 to 16 hours post pickup. And then once we get that sequencing data, we're analyzing it on the CHOP Linux-based computing system. And so in total, this pipeline costs about $28 per reaction and delivers next day results. And the bulk of today's talk will be focusing on our pilot result analysis of these retrospective viral samples, and then I'll have a few vignettes of the new prospective sequencing. So just to go over our clinical data, so we have 525 tests that we're including in this retrospective sequence analysis. They come from 462 patients. Seasonality, nothing is terribly exciting. It makes the most sense that we see the biggest amount of norovirus in February and January. March is still a pretty big month, and so we're hopeful that after March it'll drop off, and this really is a disease that is most common in the fall to winter months. Being very new to pediatric microbiology, it was really fascinating to actually see that when we take the age of the patients that are positive for norovirus G2 at our hospital, about 50% of them are toddlers and preschoolers. So I think any parents of young children will recognize that this is a disease that is often coming from the daycare setting. And then about equal amounts are coming from infants, so 0 to 11 months, and school-age kids, with lower amounts of individuals coming from the adolescent and adult population at CHOP. For the most part, about 70% of our samples are being tested from inpatient settings. And just because the patient is inpatient and they are positive for the virus does not necessarily mean that it is a hospital-acquired infection. For instance, if a child presents to the ED and the child is sick appearing, the ED may admit the child and then test them once they're admitted rather than testing them in the ED. And so that would still be considered a community-acquired case. About 20% of the kids are getting tested in the ED, and then 10% are getting tested in the outpatient setting. So 2 individuals had 4 tests performed, 3 individuals had 3 tests, and then 19 individuals had 2 tests performed. And so for the most part, these are not repeat patients, but we're able to do a deep dive analysis on these repeat patients at some point. So now getting into the results of our assay, the very first thing we really want to look at is, does this work? And so we took essentially the 500 or so samples that were sent to Plasmidsaurus for sequencing, and we divided them into whether or not they produced a nice assembly or whether or not they failed. And we got this graph. So I think it's pretty apparent to see that this is largely driven by the cycle threshold or CT value, which is a proxy for how much virus is in the sample and is logarithmic based. And so around a CT value of 30, you can really see that our odds of it failing are a lot higher than our odds of it succeeding. And then there still are some sporadic failures occurring at these low CT values, but for the most part, we're able to get the low ones sequenced very well. And so the average CT value of a sample that failed was 28 compared to 21 for samples that did work. Essentially just means going forward for our prospective sequencing for IPC, we will attempt to do sequencing on every sample. We might attempt 2 times if there's a compelling reason, but just due to a limitation of the assay, if we're needing a lot of whole length RNA, it may not work. We didn't come up with this idea. It came from this really great paper from our colleagues up north at Public Health Canada, where they were able to use these genome spanning primers for norovirus G2 to perform whole length PCR and then whole genome sequencing. What we did do was improve and expand on their numbers a bit. So in this original manuscript, this was primarily using Invitrogen supplies. They tested 57 samples and 39 worked. And in our expanded effort, we test about 578 samples and 464 work using these QuantaBio supplies. And really the biggest difference and what makes it possible that my dream is to do a same-day turnaround time is that the PCR protocols were far, far lower for the QuantaBio assay compared to the Invitrogen steps, primarily because this was amplifying 1 min per kb, which for a 7.5 kb virus is going to take a bunch of time when you want to also have 35 to 40 cycles. So we're really fortunate that the assay both improved the percent of samples that were sequenceable and that did it in an actionable time frame for us to give results to our colleagues in IPNC. Our sequencing pipeline proceeds from a least granular to a most granular analysis. Least granular is using this phenomenal CDC genotyping tool where we're able to get kind of a high-level view on the norovirus genotypes circulating in our community. And then we're going to dig a little bit deeper into a maximum likelihood phylogenetic tree where we're taking all these norovirus genomes and we're aligning them to see who's most closely related to whom. And then finally, we are doing very fine-grained temporal spatial single nucleotide polymorphism or SNP analysis because we're able to pair the phenomenal clinical informatic metadata with the sequencing data to really figure out where are these patients when they are testing positive and where are the samples that those viruses are most closely related to. In the interest of time, we'll primarily focus on the genotyping and then the temporal spatial SNP analysis, but I have one figure showing the maximum likelihood analysis at the end. So the very first thing we really wanted to do was just to QC our data. So we took all the norovirus G2 assemblies that we got back, and we very simply just looked, how big were they? Norovirus should be about 7,500 base pairs. There is a polyadenylation site in the genome at the very end of the genome, and so we expect to see some variation in size. And so we were pretty happy when we made this plot, which shows the size distribution of all of the retrospective samples. And we found that for the most part, it is exactly what we expect, where our mean plus or minus 1 standard deviation falls within 7,520 to 7,600. There's a few genomes that look a little bit smaller than would be expected and a few that are a little bit larger, but for the most part, they all are the expected size. So we're really happy with the quality of the assemblies, and that gave us confidence in our ability to interpret these results in an actionable way to help our patients. Now, moving to the genotyping. Being unfamiliar to norovirus genotyping was kind of a confusing scheme to learn at first, so hopefully I can summarize it efficiently, but it's a premise is on dual classification, and it used to proceed based off analysis of a 300 base pair region in the VP1 gene, which is that capsid, and then the RDRP, which is the polymerase gene sequences. And so in the VP1 genotyping scheme, that's where we get these large genogroups. And so all of the genogroups that we are analyzing will be G2. In norovirus, there are 10 genogroups denoted by these Roman numerals. Some infect humans, some do not affect humans. And then in addition, the VP1 genotyping also gives us information on a specific type, which is devised by number, and then also the location of where that sample was first identified. So a common one you'll see is G2.4 Sydney strains. And then finally, the RDRP typing, due to the ability for norovirus to recombine with similar strains, you can get mismatching of different capsid and polymerase chimeric viruses. And so this RDRP typing refers to just the P segment and it'll also give you a number. And putting that together, you get results like this, which show the most common genotypes identified in our retrospective collection. So this example using G2.4 Sydney P16 was because that particular genotype is by far the most common genotype that we see at CHOP. We see a smattering of other genotypes, so shown in colors are the top 9, but in total, we actually identified 30 different genotypes, which was really interesting to see. Being very new to norovirus genomics, I then also wanted to learn sort of, is this representative of what we see globally? And thankfully it is. The G2.4 Sydney P16 strain emerged from Australia around 2012 and very quickly became the dominant clone that hospitals are likely to see internationally. And so it makes sense that what we see most common at CHOP is what we see most common out in the world. Since we have the clinical informatic metadata, we are then also able to go into the seasonality of these samples. And so we can see, did the norovirus genotypes differ each year, or are they, for the most part, the same? And what we see is really both of those things are occurring. So when we add on the different years, we can see that that bottom red bar, which is the stacked bar plot relating to the proportion of samples that are the different genotypes. This bottom red bar is the G2.4 Sydney P16 strain. And so overall, it's 37% of our samples. And you can see that for the most part, it's pretty consistent as to be about 30 to 40% of the total samples, and is always going to be the most dominant genotype per year. So we see some swapping in and swapping out. So for instance, in 2024, G217P17 had a really large amount of samples, even though it was lower in these previous years. And then in 2020, this G6P7 had a really big year, albeit in a much lower amount of samples due to the COVID pandemic, and then has been consistently present, but not nearly to the same degree as this initial sample. And so to us, it indicates that there's really a pressing need to identify these other genotypes in addition to the Sydney strain, because they still can cause the hospital-borne outbreaks that we're very interested in stopping. So now I'm moving into SNP analysis. Really what we're doing is we're just basically taking two sequences and then we're just counting, we're aligning them, and we're counting the number of single nucleotide polymorphisms. And so in this example of a virus A and virus B, we have 2 SNPs. But we're not doing this on just 2 viruses. We're doing it on about 500 genomes, and 500 pairwise interactions is 124,750 pairwise comparisons. So when we take that giant matrix of an all-by-all SNP count and we digest it into a easy-to-interpret histogram as humans, this is what we get. So towards this end of the histogram are the distances between the various genotypes. And we're very fortunate that really from an outbreak investigation, transmission dynamic study, all we really care about are the samples down here. So these are the samples that are in the most closely related bucket. They're all less than or equal to 10 SNPs to one another. And those are the ones that are likely causing or that are examples of outbreaks occurring inside of the hospital. So we next wanted to do a deep dive into just these most closely related strains. And the way I'm showing to portray that is through this cytoscape network diagram, where essentially each of these individual black dots is a norovirus genome. And a gray line connecting these black dots indicates that there's less than or 10 SNPs between any of these specific genomes. And when we do that on the entire cohort and we can see who's most closely related to, what we get are 30 pairs of closely related genomes, 14 triplets, 6 quartets, and then 7 clusters containing 5 or more genomes in each of these clusters. And for our first pass analysis, we were really interested in learning more about these specific clusters. So we did another deep dive into them. And when we look into these clusters, it was really fascinating to see that they're all temporally and spatially linked to one another. So they're all essentially occurring within a 1 to 6 week period. and the months are shown on the individual clusters. We then next overlaid what is the genotype of the viruses causing these clusters, and it really is not terribly surprising that since that Sydney P16 strain was the most common infection that we see at CHOP, it's also the one that causes most of these large clusters. So 3 out of 5 of these clusters are caused by the Sydney P16 strain. You can see that it occurred in 2024 and also 2023. This Sydney P4 strain, again, going by the norovirus genotyping rules, has the same capsid sequence as the Sydney P16, but it has a different polymerase sequence. And this one caused an outbreak in January 2022. And then this G3P12, completely unrelated to the Sydney strains, it caused an outbreak in December 2022. So just to give another example of what we're able to do with the sequencing data, let's dig even deeper into just this February 2024 outbreak caused by the G2.4 Sydney P16 strain. And again, this is a network diagram. This is a bit more crazy of a network diagram, but I really like this figure. So it's force weighted. So we essentially are telling the program to cluster values that are similar to one another and separate them from values that are far from one another. And then we colored it so that each of these individual dots represent, or each of these individual bars, again, represents a norovirus genome. Here we're actually showing our study ID that we use to de-identify and send out the samples for sequencing. Red lines connecting any of these 2 black dots indicate that there's 0 SNPs. They're exactly the same virus as one another. Blue lines indicate there is 1 single nucleotide polymorphism. And then gray lines indicate there's 2 through 10 SNPs. So that's what I want you to keep in mind for both this figure and the next figure, is that the maximum genetic distance within this network and tree is just 10 SNPs. And so doing this is really fascinating because we can see, for instance, up here, there's this group of 5 strains that all, for the most part, share 0 SNPs with one another. There's this triad of 3 strains down here that all share 0 SNPs. And there's a pair in here that all share 0 SNPs. But it's really fascinating that these are all related to one another because there's only 1 SNP separating these samples from each other. And so this is indicating that there's really a network of very closely related isolates occurring inside of our hospital. And we next wanted to make a tree and overlay the time and the location information. And when we did that, it was really fascinating. So we're starting with just the samples that were recovered from ICU A, and the day value relates to how many days post the oldest sample these individuals came from. So we're seeing that one of this group contains all 5 of those samples that have 0 SNPs to one another, and they're all in the exact same ICU. And what was really fascinating is that the time span of these infections. So the first infection that's occurring in this ICU was on day 13 of the outbreak, but 10 days later, we still have a patient who's infected by the same exact virus. And then when we add in some strains coming from different ICUs, it was really fascinating to see that there was actually transmission between the ICUs. So that triad that I mentioned in the network diagram is down here. It contains these 3 individual genomes. 2 of them are coming from ICU B, but then one of them also was found in ICU C. And what's really notable is this distance from the tree for the sample from day 30 compared to sample from day 9. And so again, there's 0 SNPs between this R451 and this R499, but they're separated by 21 days, which really shows that the norovirus is really good at living on a surface and evading our cleaning efforts. It was also really interesting to see that there were 2 samples from the emergency department that were lumped into this cluster. This sample, R437, is an outgroup to the rest of the phylogenetic tree, and so it can be considered the most distantly related one. And this sample, R456, is an outgroup relative to these strains, but not to these ones. And so my interpretation of this is that there is still community circulation of these strains. It would be really fascinating if I was a public health laboratory to be able to do more in-depth contact tracing to determine, was there any social or non-hospital connection between the two kiddos that presented to the ED and the individuals who are hospitalized who are getting the virus, but that still remains to be seen. So overall, it's shown, though, that this technique enables us to really get nitty-gritty, fine-grained analysis on how the samples are all related to one another and where they're occurring infections and at what time. And that was just an analysis on the big clusters that we were looking at. We're really interested in identifying what we're calling this cryptic transmission, where we could see duplexes or triplets. And these represent opportunities from an infection control standpoint to intervene to prevent outbreaks from occurring at a small juncture before they get really big. So now switching gears to perspective. So our study IDs follow very simple nomenclature. It's an R for retrospective. It's a P for perspective. We sequenced about 85 perspective samples, I believe, so far. And when we take this phylogenetic tree and we can identify how closely related they are to one another, we're able to essentially group them into 3 groups of 0 SNPs. So this very large group actually represents a hospital outbreak that had occurred 3 weeks ago that we were readily able to use the sequencing data to determine that it was a bona fide nosocomial transmission event. And then we're fortunate that the infection prevention and control measures were able to stop the outbreak from occurring. And also this outbreak remained confined to an individual ICU. And then we're going to zoom in on just this vignette of these 2 very closely related samples that have 0 SNPs to one another. So this story starts with patient A presenting to the CHOP ED. This is winter 2025-24 with gastroenteritis. The patient A is relatively healthy outside of this bout of gastroenteritis. They order norovirus PCR in the ED, and then that patient was discharged. Patient B is very medically complicated, and patient B presents to the ED for another infectious disease related issue. They have a JG2 site infection that we ended up culturing Pseudomonas aeruginosa, and due to that site infection, they are presented to the hospital, and then they were eventually admitted. Before they're admitted, they sat down in the same room in the ED that patient A was just in. The next day, the IDDL was able to result that the patient A sample was positive for norovirus. And then 3 days later, after patient B was admitted for IV antibiotics, patient B develops gastroenteritis inside of the hospital. The norovirus PCR is ordered, and that same day is resulted as positive. And then 2 days later, we get back our sequencing data, and we were able to learn that there was 0 SNPs between the day 0 sample from patient A and then the day 3 sample of patient B. And we're fortunate that because we have such great colleagues in infection prevention and control, once we relayed them that these 2 genomes are exactly the same, they were able to do this epidemiology linking, and it took them 20 minutes to figure out that patient A and patient B overlapped in the same room in the emergency department. And so in terms of using the sequencing data to change hospital practice, does it mean that the ED should use their norovirus cleaning protocol during all cases of gastroenteritis during the winter, because we've shown that this transmission might be able to occur, and there's a different cleaning protocol for individuals who are PCR positive for norovirus compared to other infections. And so even though it might be more laborious and more expensive because we're using different cleaning products, is it worth it to, in the ED, just do it for all cases of gastroenteritis because the background rate of norovirus is so high, and because we are now able to determine that these 2 infections are really closely linked? So in conclusion, we've shown that whole genome sequencing of norovirus G2 is cost effective, it's relatively quick. We have an 80% success rate of our assembly on the first attempt with a strong dependence on CT value. That Sydney P6T strain is the most common genotype we see at CHOP, with other genotypes varying annually. And the SNP distance is really phenomenal at showing clusters of highly related genomes spreading within the hospital. We have a lot of current efforts that we're working to expand this program. So we are continuously sequencing new samples for the hospital. We're doing runs about every Tuesday and every Friday. Our goal is to eventually do that every single day. We're figuring out how we're using this next generation sequencing data to target infection prevention and control interventions, such as hand hygiene reminders, bleaching rooms. This is, I really think of as kind of the beta phase of our algorithm, and we're working on phase 1 of training the IDDL laboratory staff on the protocol so it can be part of our normal molecular shift schedule. And then the Center for Microbial Medicine, fortunately, now has a MinION. We're working on getting that MinION operational so that we can really hit a same-day turnaround time on the sequencing data. And then eventually, we'd like to validate it in a CLIA-approved manner, interface the results with LIS. And then I'm actively working on how I can upload the sequencing data to the CDC CaliciNet surveillance network so that it might be incorporated into a national surveillance program for looking at norovirus outbreaks. And all this really exciting, fun work, and me being able to talk to you all for 45 minutes would not be possible without a phenomenal amount of great staff at CHOP who contributed to this effort. Notably, my 2 co-directors, Rebecca Harris and KP Smith, the laboratory leadership, Kara Thompson, Jackie Bazzoli, and then the phenomenal work of our laboratory staff, Jeff Fink, Mike Elkin, and Jess Llewellyn, all the bench level staffs for finding the samples and for processing them if we need them re-extracted. This work is occurring under Pathogen Registry, IRB, and I'd also like to thank our colleagues in infectious disease and IPC, notably Erica Hayes, Logan Grimes, Hee-Wan Yoon, and my colleagues in the Center for Microbial Medicine, Ahmed Mustafa, Paul Planet, and Joe Zacular. And then this is my email if you have any follow-up questions. And then if we have time, happy to answer any questions. And again, thank you all for your attendance.
Thank you, Dr. Potter, for such a great talk. And I do have a couple questions that we already received. So the first one is, some failures occurred with samples with higher CQ values, likely due to the requirement of full-length RNA for the ONT assay. Do you think that downstream short read sequencing, such as Illumina sequencing, would improve the success rate?
Yes, that is a very good question. And I think the short answer is yes. The long answer is yes, and it's already done. So in this original paper from Public Health Canada, they did Illumina and Nanopore, and the results shown here for their Nanopore sequencing, but they were able to take some of the samples that failed Nanopore and then did Illumina sequencing, and it worked. And I think it's exactly what you're thinking of, which is that this assay relies on the full length of the virus. And so you need enough of the full length. And if you even have enough samples, maybe the ones down here, we are kind of conjecturing there's enough virus, but RNA is a very finicky macromolecule, and so it could be degraded, and so we're not able to get that full length amplification. So I think Illumina sequencing would then offer an alternative because you're able to get that short read amplification. And it's a 7.5 kb genome, so I really don't think you need that much additional depth. The reason we're sticking with Nanopore is just that the ability to analyze the reads as they come directly off the machine is what really fascinates us and what makes us want to have a same-day turnaround test.
Our second question, and it was mentioned that the data has been used to suggest that norovirus cleaning protocols should be implemented throughout the winter. What is the long-term actionable goal for spatiotemporal identification, such as quarantine, room floors, bringing in additional cleaning protocols, temporarily notifying patients, etc.?
Yeah, that is a great question, and it's one that in the beginning of this project, I was thinking about it a lot because I knew I'm good at sequencing stuff, and I'm good at when you sequence stuff and you can make trees of anything. And so it's what are we going to do with that sequencing data that then provides additional information compared to what was IPC doing before? And after a lot of really good discussions with our colleagues in IPC, what I think we've really learned is sort of the IPC thinks of the hospital in very like horizontal way where everything are built off of the ward location, but we've shown really that norovirus does not care which room or which part of the hospital the patient is in. It is able to transmit through the hospital, and we're still figuring out on what are these possible vectors of how norovirus can go from ICU B to ICU C. And so in terms of like the interventions, yeah, I think a big one is the room cleaning because the norovirus environmental decontamination protocol is just more in depth compared to just the normal patient discharge. And so it could be that maybe it's not economical to do it for everyone in the ED, but maybe we can find what are characteristics of patients who are likely to have a norovirus. And so we can know, all right, we don't need to do it for everybody, but maybe we could do it for just this small subsection. And then in terms of stuff also in the hospital, we're really interested in identifying, at least it's helpful for telling IPC two things are unrelated, even if they came from the same patient. And that was helpful 2 weeks ago. We had, I'm not sure on the exact day, but maybe we'll say it's a Monday, we have ICU X, and then Tuesday we have ICU X again, but they're two separate genotypes. And so, well, before IPC could be worried about this as a cluster, we know that these are completely unrelated virus, and so there's really no issue. So yeah, the intervention part, we're still working on it, and it's exciting because this is sort of a new frontier that I like to think of for hospital epidemiology. So if there's any ideas, feel free to contact us because yeah, very open to how this data might be helpful.
Next question, we have, what is the mutational frequency of norovirus for environmental adaptation? How is the result affected by the cDNA primer design?
That is a great question. So you can tell from this tree that there's really not like that much of a genetic distance between this guy and this. So these are actually like 0 to 1 SNPs. But even from here, from like P061 to the end of this branch, P012, that's still only like less than 100 or so SNPs. And so there is literature using Bayesian phylogenetic analysis to essentially create a timestamp on how many mutations to expect in the genome per year. And we've used that initial estimations for our data set, but when you basically take that, and that's primarily based off amplicon sequencing, and then when you scale it up to whole genomes, the calculation that we got was about 34 SNPs per year is what would be considered the same strain, which just feels, which I know is a very unscientific word to use, it feels like it's too big, really. And so that's something that we are interested in doing is now that we have so many whole genomes and we do have the timestamp information, at least going back the last 5 years, we could get an accurate information on what is the kind of molecular clock for norovirus. It's not our highest priority because I like to sort of rein in my scientific brain to say this is a transmission study, not an evolution study, but we are very interested in learning like how much is it evolving, where in the genome is it evolving. You'd expect the capsid, but maybe we see some polymerase mutations. But we do at least conjecture that for cases like this is like, and that's why we think that this is kind of environmental habitation, because in the environment, norovirus should not be accruing mutations. It should only be accruing mutations when it's infecting cells and has its error-prone RNA polymerase. And so you would not expect something, if patient A months later infected patient B, there still should be some SNPs. The fact that we have two strains that are 21 days apart but are 0 SNPs to us indicate that it's the same virus. It's just sitting somewhere on a surface waiting for an unfortunate child to make contact with it where then it can infect that child's GI tract. So yeah, the kind of molecular clock of it, very interested in from a basic science standpoint. Right now we're saying 10 was our cutoff to kind of create this analysis, but really it looks to be pretty binary of 0 to like 1 SNP, kind of comes up with the same information. And so I'm very curious to adapt that Bayesian analysis. It's kind of a weak point in my training to then get a more accurate cutoff eventually.
And I have another technical question. Do you have any information regarding sample failures based on genogroup or genotype? Have you tried Sanger sequencing on samples you could not get full-length transcripts?
That is another good question, and really no. Essentially, for the ones that failed, we did, so it says failed first round. Some of these samples, we essentially loaded as much as we can. Like I forget, I think our actual protocol had like 5 microliters of RNA as input, and then the long PCR had 5 microliters of the Q script as input. And so for the samples that did fail, what we did was just max out the reaction. So we doubled the RNA and we doubled the cDNA input, and it really did not make that big of a difference. I think 30 of the 96 that failed that we tried that sort of massive amount sequencing on ended up working, but really it was not a lot to convince us that it's worthwhile to do periodically. And we think that that's just because although we're doubling the amount of data, the CT value is a log scale. And so it's just at a certain point, you're running against the limitation of how much virus is in the sample. But I'm not sure, I'm not as knowledgeable for Sanger sequencing to know if that would have better sensitivity compared to what was brought up earlier for the Illumina sequencing method. There's definite utility in, optimizing that protocol even further, using Illumina for cases where it did not work the first 2 times on Nanopore. It's just not something that we've really tried to do for this particular cohort.
Next question, is there any information or theory on what makes the norovirus capsid so strong in protection from external effectors and cleaning measures?
Oh man, that's a great question. And I am not the person to answer that. But I did training Washington University School of Medicine. Was very fortunate to have the home of Dr. Skip Virgin for about over 20 years, who's a really expert in norovirus and trained many of the great people in the field. And so I'm sure there's literature out there that determines why is the capsid so strong compared to, so in virology, I often, at least in my head, think of envelope viruses like HIV, hepatitis C, very weak on surfaces and will often die immediately or just remain non-infectious. Non-enveloped viruses such as norovirus and other viruses like rotavirus are generally more resistant because they don't have that lipid envelope. But what makes norovirus so unique, even compared to the other gastroenteritis viruses, I'm really not sure, but it'd be really interesting to see kind of, if you can engineer the capsid in a way to figure out like these mutations make it weak and these mutations make it strong. I think that'd be really cool. And so, really interested in maybe phase 2 of the study to look more at the evolution of where are these SNPs occurring in the norovirus genome and what can that tell us about how these strains are infecting kids.
And it looks like this might be the last question we have. The fail rate you reported was on retrospective samples. What fail rate are you seeing with prospective samples? Recognize the numbers are smaller at this point in time.
This is a great question, and I didn't show it because the answer is that the fail rate is slightly lower, and I'm not sure exactly why, but it's about like 75%. So it's in between these two, and we've yet to really do the full grain analysis for the prospective samples to figure out why is our fail rate a little bit higher than the retrospective samples. Our conjecture is that for the retrospective samples, the IDDL had saved the positive stool for all these samples. And it's not in our clinical protocol, but in our research protocol, we're spinning down the stool to separate the fecal material from the supernatant where the virus is. Whereas for the IDDL assay, we want the bacteria. We run a similar analogous molecular platform for the most common GI bacterial pathogens. And so we don't have that centrifugation step because we need the bacteria clinically. And so our sort of initial hand-waving conjecture is that the retrospective samples, the starting RNA input is a bit cleaner compared to the prospective samples. And so it could be for some of the prospective ones, if we're really, really interested in figuring out what that genotype is, we might need to do extraction, and we might need to do that centrifugation and extraction. The one thing that's nice about not doing that is that this cost then is already eaten in to our whole genome sequencing pipeline because we're starting with the RNA extract that the IDDL already has and was going to throw away until we said, please give it to us. If we were to do that cleanup protocol, we'd have to re-extract, and so the samples would just get a bit more expensive. But that is kind of our conjecture for why we see this a bit of a dip. Not terrible, but still lower than our rates of the retrospective sequencing.
And with that, we'll wrap up our Q&A session. Thank you all for joining us today, and a special thank you to Dr. Potter for sharing his expertise and insights. We hope you found today's webinar informative, and if you have further questions or would like to connect with Dr. Potter, please feel free to reach out. And we appreciate your participation and look forward to seeing you at future webinars.